Good day! I'm Dr. T and welcome to my office. So today I'd like to talk a bit about mutations. Okay, so usually we're thinking in terms of DNA replication here. As the DNA replicates, or if it takes damage and it gets repaired, occasionally one base pair was supposed to be put in and a different one was put in. So you've got a typo. Alternatively, you've got some damage where, some of the DNA just wasn't replicated or a bunch of base pairs were added in that shouldn't have been. Okay, depending on where in your DNA depends on how bad this is. So let's talk about what happens if it's in the gene itself. If it's in like a promoter region, that can easily do other things. That's editing the metadata. But we're going to talk about in the gene region to start with. Okay, so the bases then are going to be transcribed and translated. During translation, you of course got your codons. So what can happen? Let's assume you just have a single base that's been switched. There's a classic case is sickle cell anemia where this happens. Okay, so you've got a single base that gets switched. First off, it could be a silent mutation. So a silent mutation is one that while the base is switched, so you've got a different codon, it happens to be recognized by either the same tRNA, because some tRNAs can recognize multiple codons, or it's recognized by a tRNA that has the same amino acid on it. Because, once again, multiple codons can code for the same amino acid one way or another. This is what's called a silent mutation. Yes, you have changed the genetic code. Did it matter? No, not at all. This is meaningless. This is an interesting one to start looking at things like ancestry, especially things like silent mutations. There's actually a lot of different things to look for ancestry. But as mutations build up, that implies a longer time since a shared relative. And thus, if you look at my genome and my father's genome, they'll be fairly similar. And then if you looked at my great-grandfather's, okay, a little bit different. Now if you look at my second cousins, my second or third cousins, whatever you call those folks, we're going to have, different mutations. And thus you can say, hey, we're similar, but we're probably second cousins. That kind of scenario. Okay, now, of course, that base that got switched, it could do what's called a nonsense mutation. In this case, it switches that codon from being whatever amino acid to being one of the stop codons. In this case, that protein is stopped, or that protein's production is stopped prematurely. This could easily result in a protein that just doesn't do anything. It's been chopped off. An important part is now missing. Interestingly enough, there's a immune protein that there's a mutation where part of it's missing, and those individuals with that mutation are resistant to the Black Death, so Yersinia pestis, and HIV. It's like random. So if you're of European ancestry, there's a small chance you have this particular mutation, which makes you very resistant to Yersinia pestis and AIDS. Really random. Most of the rest of the population doesn't have the mutation. It's a particularly rare one. It just happens to be Northern Europeans that typically have it. And it's like 1% at most of those. Okay, moving on. Oh, by the way, the reason it's there is because that's where the virus latches onto. Now, moving on. You could also have a missense. This is kind of like the silent mutation, but instead of changing the codon so it's the same tRNA, it's now a different one. So you have a amino acid that's put in, but it's the wrong amino acid. This is what we see in sickle cell anemia. There's one amino acid wherein the wild type, or what the wild type is kind of the generic term for the normal allele, or what we would consider typical, but normal and typical have more loaded meanings, so we call that wild type. And then the mutation on this one, the different alleles, the allele that leads toward sickle cell anemia is what's called a non-conservative missense mutation. In this case, it is going from a hydrophilic to a hydrophobic amino acid, and that causes the proteins to stick together like crazy, which is the big problem. So your hemoglobin sticks together, and that is a curse I would not wish on my worst enemy. Now, that mutation can be a non-conservative mutation where it's a radically different one. Alternatively, it can be a conservative mutation. Similar amino acids, isoleucine and leucine, if you look at their structure, they're really close. So if I switch from one to the other, good chances it's not going to make that much of a difference. We can actually use these mutations, specifically the missense ones, to understand how our proteins work. So if I take the sequence of amino acids, the primary structure of a bunch of the same protein, but from different organisms, so I could pull ones from like humans and mice, rice, assuming the plant had it, Arabidopsis, which is a small weed that we do lots of study on. It's like the botanical version of a mouse. Cows, chimpanzees, anything I want, maybe 20 or 30 different things. What I can do is I can line up, and I can have computer programs line up these sequences, do a sequence alignment, and that'll show me what sequences are conserved across a large spread. These are ones where, had a mutation occurred here, probably the protein would no longer function, and thus those individuals wouldn't have survived. Therefore, those amino acids that are conserved are potentially very important. The analogy I always liked was, imagine you're trying to figure out how your car works. You take a bolt out of the engine, nothing happens. Not that interesting of a bolt. You take a different bolt out of the engine, and the engine falls off. Apparently that bolt's important. So now you're looking at, well, okay, what are the similarities between these? Also, you're looking at, well, what amino acids are conserved? So these have conservative missense mutations. Okay, these probably need to be, say, hydrophobic or hydrophilic. They probably need to be similar, but they don't have to be spot on. And then which ones are not conserved? They're just any old thing. Well, in these cases, that's probably not an important part of the protein. You can put anything there. So I'm going to look and be very interested at the conserved ones. And I'm probably not going to care about the ones that aren't conserved because they probably don't do anything. Okay, now I can have mutations, though, that are not just swapping out one base for another. I can delete a base, or I could add a base by mistake. And these are a little bit more of like a repair oops process, but you can have it during DNA replication, etc. These are what will create what's known as a frame shift mutation. I mean, I guess in theory, if you took out 3 bases or added 3 bases, you're not going to do a frame shift. You're just going to add an amino acid right on, which does happen. Don't get me wrong. If you look at some of the sequences, you do see amino acids just chopped out in whole. But a frame shift is now where all of a sudden, because there's no punctuation, the codons, the brackets around which, here's 3, here's 3, here's 3. Well, if it's here's 3, but I chopped out 1, now it's here's 3. Now here's the first 2, and then the first one of the next one, and now you got, instead of 1, 2, 3, it's 2, 3, 1, 2, 3, 1. All your codons are now completely changed. And this is typically going to lead to a protein that does not do anywhere near close to what it's supposed to because the amino acids are completely messed up. So frame shift mutations typically are fairly devastating mutations to your protein as well. But you can also get some interesting ones where, once again, you have a whole codon dropped or a whole codon added. Or if you've got repeats, you can do some repeats. This isn't the only things you can do with mutations on kind of a larger scale. It's not uncommon to see an entire protein just get duplicated by mistake. So it's like, okay, I copied it. Did I copy it? I'll copy it. Oh, there's 2 copies. Oh, well, moving on. Or a whole protein just goes away. So that's not uncommon with DNA. A little bit more of a drastic thing. Typically, we're thinking of small, what we call point mutation, which is one nucleic acid, one base getting swapped for another. That's typically what we're dealing with. And that does, lead to some interesting, how does this protein work? The mutations do give you some clues as to where to start looking. So with that said, have a wonderful day.